Alright, so here we are for a video that goes over part 1 of chapter 2 of Biochem 1. So, I don't know if these are helpful or not, but it at least helps me to kind of talk through everything and see how things fit together, or something occurs to me that didn't occur while I was typing, that sort of thing. But if you look on Canvas, we should have 3 sets of notes for chapter number 2. It was a pretty long chapter. That being said, it's not a lot of new material. It's stuff that you've probably already talked about, if not in Gen Bio, maybe some anatomy, if you've had a class like that. So it is a lot of, water cells, what makes up cells, talking about the organelles, which, yes, it's a lot of review stuff, but also it's stuff that we need to know going forward, so we don't have to think, like, okay, what was a ribosome? Okay, what was happening? Okay, so this first video will go over the notes that are listed as part 1, and then this is what we would have gone over on Monday, and then I will make a video here in a couple minutes. Going over part 2 is what we would have gone over today. Today's Wednesday as I'm recording this. And then part 3 would be what we had gone over on Friday. I'm thinking we will pause there and then kind of have a big review over chapters 1 and 2. Maybe wait until we get through with chapter 3. Like I said, it has to do with water and a lot of more background stuff. Haven't decided yet. But we should have another quiz posted, if not today, then tomorrow or Friday. Again, not difficult, just make sure that we're all kind of following along, at least checking up on Canvas, that sort of thing. All right, so to just jump right into chapter 2, this is the cells chapter. I like talking about cells because, human bodies have lots of cells, so they do lots of fun different things. So I'm ready to go. I've got my official Braves video making shirt on. Okay. So first of all, a nice little book definition of what a cell is. They said, cells are complex, intricate molecular machines that can sense and respond to their environment, transform matter and energy, and reproduce themselves. So that's just a fancy way of saying cells are tiny living things. They're the smallest component of life. So if you think back to what we talked about in the previous chapter, we said, okay, cells, smallest unit of life, come from existing cells. All living things are made of cells. So that kind of is a way of saying that cells are living things. Okay. There are 2 broad types of cells. We have prokaryotic cells, and then we have eukaryotic cells. So prokaryotic cells, when I was in school, it was just bacteria. Okay, we said prokaryotic cells are bacteria. And then they said, well, there's bacteria, and then there's also archaebacteria. They came up with a new kingdom of organisms. So prokaryotes, bacteria, archaebacteria. So that's regular bacteria and bacteria that can live in like thermal vents in Yellowstone National Park or whatever. Good definition of prokaryotes are they are single-celled organisms and they lack a nucleus. So only one cell, little tiny one cell, and no nucleus for their DNA. The DNA is just kind of floating out there, and we'll talk a little bit more about that. Whereas eukaryotic cells, think, okay, living cells. So animals, like human beings, plants, fungi, protists, which I've never seen one in the real world, but comes up in every textbook. Okay. Eukaryotes can be single-celled or multicellular organisms. So we can have an amoeba, that's a eukaryotic cell, it's one cell. Or we can have a human being, that's trillions of different cells. Okay? And our cells all have, if there are eukaryotic cells, they have a nuclei. There's a huge exception, red blood cells, they lose their nucleus. But again, we'll talk more about that. So for definition purposes, eukaryotic cells have a nucleus. Okay, eukaryotic cells tend to be a lot larger. So, you have a big eukaryotic cell here and then a little bacteria here. Okay? Tend to be a lot larger, and they tend to be a lot more complex. So eukaryotic cells contain several organelles, and we'll talk, part 3, I think, of this chapter is talking about all of those organelles. Okay? But basically an organelle is kind of like a little organ inside the cell that performs a certain job. both prokaryotic and eukaryotic cells are similar chemically, so their cell membranes are made up of the same type of stuff. They both contain DNA as their genetic material. Okay, so they're alike, but also different. So just looking at the textbook, it made note of, here are some concepts that we need to be aware of that are related to cellular processes. And some of these seem a little textbooky, not real world stuff that we need to know, but I did want to go over it to make sure that we're all kind of on the same page. Okay? So the concepts related to the cellular process, we need to know about water, and water has its own chapter here in a little bit. We need to know about cell membranes. We need to know about how cells perform self-assembly, so they basically recreate themselves. Again, all cells come from existing cells. We need to talk about molecular machines, which I think is kind of an interesting concept that it took me a little while to wrap my brain around. And then there is signal transduction, so that's an important part of cells being able to communicate. And then lastly, the book threw in something called macromolecular crowding, which for completeness sake I've included it. Okay? So the first of those points has to do with water. So obviously water, essential to life. It dominates chemical processes within cells. Basically our bodies, they work based on where is water? Is water here? Is water there? Is it blocked out of this compartment by membrane? Does it flow through this compartment through a membrane? Okay? Again, we'll talk a lot more about water when we get to the water chapter. But the big thing to be aware of is that water is very, very polar. Okay? So we have H2O, we have our oxygen atom, we have a hydrogen atom coming off here, we have a hydrogen atom coming off here. Okay? Hydrogens can't pull electrons very strongly, oxygen can, so all of those electrons spend most of their time with the oxygen. So that oxygen tends to be very negative at all times, and those little hydrogens tend to be very positive at all times. So it has these charges, these pseudo charges on it a lot. Water is also bent, so we have oxygen and we have hydrogens. It's not hydrogen, oxygen, hydrogen in a straight line, so those charges don't cancel each other out. Okay, instead it's bent, and so we have regions of different charge. Okay, because it's polar, that means it is able to interact with a lot of different things. Okay, it's got charges. It's not neutral. It doesn't just want to hang out. It wants to interact with a lot of different things. And its interactions with different things, we kind of define substances based on how they interact with water. So substances that are hydrophilic, so water-loving, they interact well with water. Okay? So things that are hydrophilic tend to be other polar things. So things that have positive or negative charges, things that may have those pseudo charges where they don't share electrons evenly. On the flip side of that, hydrophobic or water-fearing substances don't interact very well with water. Okay, those tend to be the very neutral substances. Fats, hydrocarbons, a lot of organic things tend to not interact well with water. So, you drop salt in water, lots of charges, it breaks up nice and evenly. You drop a very nonpolar lipid in water, and it's just going to kind of congeal and keep to itself with water kind of surrounding it. It's not going to interact very well. All right? So that's point number one about water. Point number two is about membranes. We'll talk a lot more about this when we talk about the cells in particular. But membranes basically are thin sheets made up usually of lipids that they enclose the cell, they enclose the organelles, they enclose the nucleus, they make up the nucleus. They're basically the barrier of cells. Okay, so a nice book way to say that is they separate the intracellular environment from the extracellular environment. Okay, they are the line between what's inside the cell and what's outside the cell. Membranes have a lot of different jobs. Most importantly, they kind of control what goes into the cell. They control transport in or out of the cell. They play roles in identifying the cell. So membranes, they have little markers on them. So if you think about our immune system, we have to be able to recognize, okay, that's a cell of my own body. That's a bacteria. So we need to be able to identify, okay, markers on that one, don't attack. Markers on that one, do attack. So membranes, they play a role in cell identification. Membranes also play a role in signal transduction. So you may have a receptor on a cell membrane. Um, you have a ligand secreted by a different cell. It travels to the target cell. It activates that receptor and tells that cell to do something. Okay, so membranes are important in signal transduction. Most membranes are made up kind of the same general structure. Um, and I'm sure you've seen all of this before, but the phospholipid bilayer. So phospholipids, we have a hydrophilic head and a hydrophobic tails coming off of it. So the hydrophilic areas point themselves towards water. So there's water outside the cell and there's water inside the cell. So the hydrophilic heads point outside the cell and inside the cell. And then the hydrophobic tails kind of form this middle section. So again, I'm sure you've seen diagrams of all that before. But that's basically the general setup of how membranes look. Okay? Membranes are more than just those lipids, though. They have a lot of stuff stuck in them, like I talked about the receptors and things. So a lot of proteins are embedded in cell membranes. So we have different types of proteins. We say integral proteins are proteins that, okay, here is our cell membrane, the thickness, and the protein goes all the way through it. We say that's an integral protein. If we have the cell membrane here, and there's just a protein sticking off one side or just sticking off the inside, that's called a peripheral protein. Okay, integral proteins, so the ones that go all the way through, they're like channels, okay, tunnels for different things to go through, or they're carriers where they take something from this side and move it to over here, or vice versa. So integral proteins include things like channels or carrier proteins. Peripheral proteins, there might be a receptor that's stuck off, or it might be an identifying mark that's stuck off. Okay. So that's the point about membranes. All right, so I think we're on point number 3. All right. The point about self-assembly. So self-assembly for cells, that just means that cells kind of build everything that they need for themselves. So obviously, for the cell to stay alive, it needs things that are bigger than just a molecule. Okay, it needs to put those molecules together to build structures that it needs. So proteins, membranes, all kinds of things. Basically, self-assembly says that it's easy for cells to do this because either the building blocks are shaped just the right way so they can build things. Cells have certain proteins called chaperone proteins that they exist to kind of fold and shape things the way that they need to be made. Also, everything that's made in the cell comes from a blueprint, and that blueprint is the DNA. So that DNA tells the cell, everything that it needs to build. So all self-assembly means is that everything that the cell needs, it builds for itself. And if it doesn't have the parts, it gets them from somewhere, it brings them into itself, or it breaks down something else and uses that to build something. Okay. Point number 4, molecular machines. Okay. When we talk about, the cells need to do things, okay, there are biochemical reactions. So there's chemicals, reacting with this to cause a reaction. There are a lot of things in cells that actually physically have to be done. So this needs to be moved here. Like, it has to actually transport in space from this area to this area. That's a little bit more complicated than just a chemical reaction. So there are things in the cells that physically move something to somewhere else, or they physically build something, or they form a lattice work, or whatever it is to physically do something. And we call those things that perform these tasks molecular machines. So a lot of times these molecular machines are things called motor proteins. So again, when in doubt, if you don't know what does something in a cell, just assume it's a protein that does it. Okay, so motor proteins, they perform a lot of these physical jobs, and so how they do it is that their shape is perfect for it. So they bind to something, and that changes their shape a little bit, which allows a filament to move over here, and then that binds to something, and it allows a filament to move. So basically, the way I've written it in our notes is that motor proteins their movement is driven by changes in their shape due to binding and unbinding other molecules. So, if you go on YouTube and you look up molecular machines, there are tons of videos of diagrams of how you know these proteins come together and how they move and what they do. I attached one on Canvas that was the first time that I saw a molecular machine demonstrated that way, and it looks like he's walking, and it's the most hilarious thing. Okay, so I think I even titled it "Hilarious Molecular Machines Video." So if you want to know how those things kind of look actually in the cell, take a glance at that YouTube video. It's like 30 seconds long. Okay. All right, so next to last, I wanted to talk about signal transduction. Okay, so cells, okay, if it's a single-celled organism, all well and good. It does everything for itself. In our bodies, our heart cells need to be able to communicate with our lung cells, need to be able to communicate with our neurons in our brain. They need to be able to talk to one another to get our jobs done. Okay. Single-celled organisms, they also need to have some kind of communication with the outside world. So bacteria need to be able to track down their food. Okay, plant cells, they need to be able to find, okay, where is light coming from? So there has to be a way to receive these signals and interpret them and then cause something to happen, some kind of response to them. So really this process is called signal transduction. Okay. A huge point of signal transduction is understanding, okay, there are receptors and there are ligands. So a receptor is something that's on a target cell that is waiting for a particular substance to bind to it. And then the ligand is the substance wherever it came from, either from another cell or from outside the environment. The ligand is the thing that binds to that receptor and then tells that cell, okay, now it's time for you to do whatever job this receptor was triggered for. Okay, so receptors and ligands. So I've got it listed. There are 3 steps in signal transduction. So first of all, we have reception is the first step. So we have our receptor, we have our ligand. Okay, let's say target cell, and then ligand came from a cell far away. Okay, and it travels and it binds to that receptor. Okay, and that's called reception. Okay. The second step is called transduction. So this receptor, because it's bound to the ligand, is changed in some way. It might change shape. It might, if it's an enzyme itself, it might catalyze a reaction. A lot of times it might be a tyrosine kinase, and it adds a phosphorus to something else. So it changes in some way, and it causes that signal to go into the target cell that it was on. Okay. A lot of times it triggers the release of kind of an internal messenger in the target cell, and that messenger is called the second messenger. Okay, and then the third step is the response. So that second messenger that was released, okay, we have our receptor, we have our ligand, it bound, okay, that's reception. That caused the second messenger to be released inside the new cell, that's transduction. Then the response is that second messenger binds somewhere in the cell and tells whatever organelle it is to do a job. Okay, so a good example of the real world, okay, we have a target cell, okay, in our bodies. One receptor that a lot of our cells have is for insulin. Okay, so insulin comes from our pancreas. It's released into our bloodstream. It's the ligand. It binds to the insulin receptor. Okay, so that is reception. The insulin receptor is a tyrosine kinase. It phosphorylates other things, so that's what its job is. So once it binds to the ligand, it puts a phosphorus on something else. So it does transduction and activates a second messenger. Okay, and then the response, that second messenger goes to the cell and it tells glucose transporters to start taking in glucose. Okay, so that is the response to the signal. Okay, all right. So all of that was signal transduction. All right, and then lastly, for completeness' sake, macromolecular crowding. Okay. Book wanted to make a note that the interior of a cell is very crowded. There's a lot of stuff. The big macromolecules like proteins tend to be kind of all crammed together, so there is a lot of space for the little things to work. That's all that the book had to say about that. Okay, so I believe that is all I wanted to talk about for part 1 of this chapter. So part 2, we talk about what exactly are in prokaryotic cells, and then part 3, talk about eukaryotic cells. So I'm going to give my voice a rest for a couple minutes and then get that video up to you. So again, just be on the lookout for when we have a review and a quiz. I'll probably shoot you an email as well. All right, thanks.